So let's talk about DNA. Now, DNA, or deoxyribonucleic acid, is a complex molecule that contains our genetic code, or the biological instructions that makes each species unique. Now, we must understand that DNA is a crucial component for virtually all cellular beings, and yes, not just in humans, not just in animals, but even in plants, bacteria, archaea, and some viruses. Now, this molecule, along with the instructions it contains, are passed from an adult organism onto its offsprings during reproduction in a process known as heredity. And this is actually what makes DNA so important since it holds the necessary information and instructions that helps an organism develop, survive, and reproduce. So to carry out these instructions, DNA sequences must be converted into messages that can be used to produce proteins, which are the complex molecules that do most of the work in their bodies. Now, each DNA sequence that contains instructions to make a protein is known as a gene. The size of a gene may vary greatly, ranging from 1,000 bases to 1,000,000 bases in humans. Now, genes only make up about 1% of the DNA sequence, and the DNA sequence outside this 1% are involved in regulating when, how, and how much of a protein is made.
These are the materials and equipment that will be needed for the experiment. First, we have strawberries to extract the DNA from, rubbing alcohol and ice water to cool the alcohol, a funnel with a filter in it, and a clear glass to collect the filtrate, salt and a tablespoon measuring device, half a cup of water, detergent, food coloring, and a mallet. The ingredients will be mixed here in this Ziploc bag. The tops of the strawberries were cut so as to remove any unwanted material from the mashing process. The strawberries are then placed inside the Ziploc bag in preparation for the mashing process using the mallet. The purpose of this mashing is to lyse or break open the walls and membranes of the cells that constitute the fruits. The mechanical action of the mallet mashing the fruit causes lysis. The manner of mashing the fruits, as shown in the video, is important. Doing it this way prevents holes from opening in the bag. The mashing of the fruit was continued for approximately a minute until a mushy consistency was achieved. After this point, the salt, the detergent, and the water can be added. As shown in the video, a tablespoon measuring device was used to get the exact amount of salt needed. An adequate amount of detergent was added to the Ziploc bag. The amount was as low as possible to minimize the bubbling. Then, half a cup of water was added to the Ziploc bag. As much air as possible was forced out from the Ziploc bag before sealing it. This was an additional measure to minimize the amount of bubbles that would form in the mixing process. The mallet was used again, this time to mix all the components into one combined slurry. The salt and the detergent each have a specific purpose. The salt neutralizes charged areas. The negatively charged phosphate groups will make DNA molecules repel each other. The salt provides sodium ions that neutralize this effect. The detergent will chemically lyse the membranes. Since cell and nuclear membranes are made of amphipathic phospholipids, the nonpolar portion of the detergent molecules will bind to this nonpolar region, and the polar region of these detergent molecules will bind to water, and the action of this will pull the phospholipid bilayers of the cells apart, chemically lysing them. The bottom of the Ziploc bag was cut, and the contents inside were poured into a funnel that had a coffee filter. This would separate the solid and liquid parts of this mixture. In the filtrate below will be the lye cells, the sodium ions, the detergent, the water, and everything else, except the solid portions of the strawberries. After a while, a sufficient amount of filtrate had collected in the glass container. As per the instructions, an equal volume of ice-cold rubbing alcohol was superimposed into the liquid container. This was done slowly by pouring the alcohol down the sides of the glass container. 2 layers formed, one polar and one nonpolar. The isopropyl alcohol is miscible with water. In order to release DNA, the histones that the DNA are wrapped around must be denatured. This is the purpose of the isopropyl alcohol. The isopropyl alcohol also has another function. It precipitates the DNA in alcohol and makes it a solid visible mass. Before the addition of the alcohol, the DNA was dissolved in an aqueous or water solution. This portion of the video is a time lapse of the DNA precipitating over a period of a few minutes.